Hello students, once again welcome to Bholanathacademy. In this video students, we are going to see nucleic acid. Okay students, sometimes in examination they are asking what are nucleic acid. So this way students you have to represent the answer. Nucleic acid students, what are nucleic acid? Nucleic acids, they are present when you see in plants and animals cell students at the nucleus. Genetic materials are there. They are nothing but they we called as nucleic acid. What is the function of nucleic acid students? They transfer genetic information. This is the main work of the nucleic acids. Okay students, now how many types students nucleic acids? Because since they are acids, there are two types: ribonucleic acid and deoxyribonucleic acid. Ribonucleic acid we can say RNA. In short, we are saying RNA. And deoxyribonucleic acid, there is a one oxygen is less, that is why the name is deoxyribonucleic acid. So short name is DNA. Okay students, so nucleic acids are two types, RNA and DNA. RNA, they are mostly present in cytoplasm. DNA, all are present in nucleus of the plant and animal cell. Now students, what are nucleic acid? Nucleic acids are unbranched polymer of repeating monomer called nucleotide. Okay students, so in examination they are asking what are nucleic acid. So this is the answer students. Nucleic acids are unbranched polymer. Unbranched, suppose this is the polymer students. So the nucleotide are like this, there is no branching. So that is why we called as unbranched polymer. Here this is the nucleotide as act as a monomer. How these monomers are combining and they are forming polymer. So nucleic acids are unbranched polymer of repeating. See students, nucleotide is repeating one after the another. That is n in number. That is n number of nucleotide will form the polynucleotide chain that is called as nucleic acid. Understanding students? So nucleic acid have a polynucleotide structure. So second definition is also correct students. Nucleic acid have a polynucleotide structure. So here I have represented students. This circle is nothing but monomer of nucleotide, and I have written here is what n. When they are coming, these monomers combines, n number of monomers combines, and they will form the polynucleotide. That is nothing but nucleic acid. Nucleotide, what are nucleotide? Nucleotide students in RNA, when you see in RNA students, they are few in thousand. Nucleotides are few in thousand in RNA, and nucleotides in DNA students you will find millions. Number of nucleotide in DNA will be what? In millions. Nucleotide containing phosphate group PO4 2 minus, that is called as phosphate group students. Nitrogenous bases, that is nitrogen containing bases, we call as what? Nitrogenous bases. And nucleotide is also made up of monosaccharide, that is sugar. Okay students, so sugar will be what? Monosaccharide. We have disaccharide, trisaccharide, only the monosaccharide it is there. So nucleotide is made up of phosphate, nitrogenous bases and monosaccharide. Understanding students? Here why it is called nitrogen? Here you will find nitrogen. There is large number of nitrogen atoms will be there. Nitrogen containing base will be what? Purines and pyrimidine. Nitrogen which are present in nucleotide students, there are two types, that is purine and pyrimidine. Purine is what? It is fused structure. They are nothing but adenine and guanine. Pyrimidine consists of cytosine, uracil and thymine. So first we will see the structure of purine students. Purine, this is the common structure of purine students. So the pentagonal ring is fused with the hexagonal ring containing two nitrogen like this. So this is the structure of purine. Now these purines is classified into two categories, adenine and guanine. In short we can say AG. Okay students, now in adenine students, what will be the possible structure students when you are going to see adenine structure students? Adenine structure, so first you draw the purines. So this is the basic structure students. First draw the basic structure of purines. So students, this is the structure of purines when we call as adenine. This when the purine ring we called as adenine, there is a nitrogen. When a nitrogen of ammonia is attached to this ring, then this purine is called as adenine. Short we can say A. Okay students, when this purines is called as guanine students, when there is a double bond here, so this bond will be transferred to here. So this will be the carbonyl group in guanine, and this is the amine. Nitrogen of the amine is connected to this carbon. So understand, this will be the structure of guanine. Now come to the cytosine structure students. This is the You can, in short way, you can learn like this: CUT. You can say cut. Okay, students. Now pyrimidine structure, students, it is a 6-membered ring containing 2 nitrogen. 6-membered ring will be like this, students, containing 2 nitrogen. Okay, students. So this is called as pyrimidine. Understanding, students? If this is the carbon number 1, this will be the 2. This, if you are starting the numbering, this nitrogen will containing first. This will be the second, 3, 4, 5 and 6. Okay, students. Now if you want to draw the structure of cytosine, what will be the possible structure of cytosine? So first you draw the pyrimidine ring first, students. If you want to draw the cytosine structure, students, here will be the NH2, which is attached to the 4th carbon, and there will be the carbonyl. Now complete the other valency, students. So this is the structure of cytosine. Okay, students. Now second structure, students. Okay, students, this is the pyrimidine, basic skeleton of the pyrimidine. Now if you want to draw the uracil, so understanding, students, here the 2 carbonyl. Understanding, students? Now complete other with valency. So this will be the structure of uracil. Now come to the thymine structure, students. How to draw the thymine structure? Again, students, you have to draw the pyrimidine skeleton first. So this way, students, you have to draw the pyrimidine structure. Now if you want to draw the thymine, here will be the carbonyl and here will be the methyl group, CH3. Okay, students. Other valency will be like this. So this is the structure of thymine. So this way I have explained the structure, students. This way you can identify the structure, which structure is the cytosine, which one is the uracil, and which one is the thymine. Understanding, students? So if you do the practice, you can learn the structure, and this is also called as G. So students, second part, that is monosaccharide. What are the monosaccharide? It is the sugar containing 5-membered ring, that is C5S10O5. Understanding, students? 5-membered ring monosaccharide we are using in the nucleotide. Nucleotide of both RNA and DNA contain 5-membered ring, that we call as furanose. Okay, students, it is a type of sugar. Now sugar here, furanose, if they are present, if sugar is present in RNA, that is D-ribose. If it is present in DNA, if the sugar is present in DNA, we call as deoxyribose. So here, students, we can see the structure of sugar which are present in RNA. In RNA, with the sugar which in monosaccharide is present, that is called as D-ribose. D-ribose. D-ribose, the sugar which is present in the RNA, the structure will be like this, students, 5-membered ring. So this way, students, you can draw the structure of the D-ribose. So carbon is tetravalent, so this way you have to draw the tetravalency of the carbon. Okay, students, because it is dextroribose, so here the hydroxyl group will be above and hydrogen will be what? Below. First carbon, in first carbon, students, hydroxyl group will be upside and hydrogen will be at downside. This is carbon number 2, 3, 4, and the 5 carbon will be like this, CH2OH. This is the 5th carbon. Okay, students. Now when we fill the hydrogen, because all sugar are polyhydroxy of aldehyde and ketone, so when you draw the hydroxyl structure here, students, the hydrogen will be above and the hydroxyl will be below. Here also in 3rd carbon, hydrogen will be above and hydroxyl will be below. Hydroxyl group will be below, hydrogen will be below, and this CH2OH will be above. So this is the structure of D-ribose. This is the sugar which are present in RNA. There is a removal of oxygen from this D-ribose, we call as deoxyribose nucleic acid. Okay, students, now we are going to draw 2-deoxy D-ribose. Okay, students, so first you draw the structure of D-ribose, which I already have drawn. So this is the possible structure of D-ribose, students. 5-membered ring, you have to draw like this. This is the carbon number 1, 2, 3, 4 and 5 will be here, students, CH2OH. So this is the carbon number 1, 2, 3, 4 and this is the 5 one. Now, students, because it is a D-ribose, so hydroxyl will be above and hydrogen will be below. Now at the carbon number 2, there is a removal of oxygen. One oxygen is less, that is why it is called as 2-deoxy. At carbon number 2, there is a oxygen is what? Going to be removed, so only the hydrogen will be there. Okay, students, because of that, the name of this sugar is deoxyribose nucleic. Other part will be as it is, students. So this is the sugar, monosaccharide sugar containing 5-membered ring, because there is a less number of oxygen atom. One oxygen atom is missing here, that is why it is called as deoxyribose nucleic acid. Deoxyribose sugar, they are mostly present in DNA. DNA will always contain deoxy ribose type of sugar called as nucleoside. Nucleoside students, what are nucleoside? Nucleoside containing sugar as well as what nitrogenous base. These two components are present. When they are present, we call as nucleoside. Mostly they are present in RNA. They are present in RNA. Okay students, so this is called as nucleoside. Pentose sugar and nitrogenous base. Pentose sugar, that is C5H10O5, and nitrogenous bases, you know. So here I have taken students, first I have to draw the structure of sugar, pentose sugars, C5H10O5. So this is the structure of sugar monosaccharide. OH will be above, hydrogen below, H, OH, CH2, OH, and hydrogen will be below. So this is the pentose sugar students. Now when combines with nitrogenous base, this is the sugar students. Now we are taking the nitrogenous base cytosine. We are represented with capital C, that is cytosine. Cytosine, it is a 6-membered ring students containing 2 nitrogen like this. So this is the structure of cytosine students. Here will be the hydrogen. Now what happens students, when these two, sugar and cytosine, will combine and they will form the structure like this students. So this is called as ribonucleoside. This is called as ribonucleoside. So nucleoside are two types students. It's called as what? Ribonucleoside. Okay students, what happens students when the sugar, this is D-ribose sugar combining with cytosine and it is forming a ribonucleoside. Understanding students? So this way I have drawn. Now if this is deoxyribose students, it will form deoxyribonucleoside. Understanding students? When the structure, when the sugar is deoxy like this students, if this oxygen atom will be not there, we call as deoxy. There is a removal of oxygen, we called as deoxyribose. See here students, one oxygen atom is missing, that is why this sugar is what? Deoxyribose sugar. Now this sugar again will combines with the purines, and now here I have taken the pyrimidine. Now here I am taking purines. In purines I am going to take adenine, that we are showing with capital A. Adenine structure students, you have to start like this. This way you have to draw the adenine structure. First you draw double bond, then you draw the nitrogen atom, then join with carbon. Okay students, now here again you have to draw the 6-membered ring like this. This way students, you have to draw the structure of adenine. Understanding students? Very simple structure I have drawn. So this is the deoxyribose. When combines with adenine, this is the sugar students. Now when it will combines, it will form the deoxyribonucleoside like this students. Structure it will form. Now this is the hydrogen. Now this hydrogen will combines with this hydroxyl group. There is a removal of water molecules. Now what happens students? Again you draw the structure like this. So this way students, you can draw the structure of adenine. Only hydrogen students, oxygen you have to not draw because it is deoxyribose. So HOH will be here, the HOH, CH2, OH and hydrogen. So this is called as deoxyribonucleoside. So mostly these nucleosides are present in DNA. So this nucleoside, deoxyribonucleosides, mostly present in DNA, and ribonucleoside, they are present in RNA. Understanding students? Now here students, hydrogen of this nitrogen will combine with hydroxyl of the first carbon. Okay students, when you number the cytosine, this is the first, so you can say 1-1 combination. 1 combines with 1. Here students, hydroxyl of the first carbon with combined with 9th hydrogen of this adenine, and it will form the 1-9 combination. 1-9 here 1-1. So this is the 1 is for sugar, and this is the number of sugar. Nucleotide students, it is containing, I already told what is nucleic acid. It is a polymer of nucleotide. Understanding students? Now this nucleotide is containing phosphate, pentose sugar, that is C5H10O5, and phosphoric acid, that is PO4 2 minus, and nitrogenous base you already know, purine and pyrimidine. Okay students, so these are the nitrogenous base. So students, nucleotide is made up of pentose sugar, C5, phosphoric acid and nitrogenous base. So what happens students, when there is a pentose sugar students, nucleotide is also two type. If it is AMP, adenine combines with monophosphate. AMP, that is called as what? Adenine monophosphate. How you are going to draw the structure of adenine monophosphate students? Monophosphate. So this is the sugar structure students. This is the sugar which is combining with the adenine structure. So now the name is AMP, that is adenosine monophosphate. Because of this adenine, it is called as adenine monophosphate. Only one phosphate group is attached to this, that is why it is called as what? Monophosphate. Okay students, H, OH, H, OH, and here students CH2O combining with phosphate group, that is why it is called as adenosine monophosphate. When it is called as deoxy cytosine monophosphate, C stands for what? Cytosine, D cytosine monophosphate. So here the sugar students, there is a removal of one oxygen from here, so this is the difference students. Now at the carbon number 2 there is a one oxygen atom will be not there, that is why it's deoxy cytosine monophosphate. So this is the structure of cytosine students. Here I have drawn cytosine. This cytosine when combines with sugar we call as, and it is called as cytosine monophosphate. Only one phosphate group is going to be attached here students, CH2O. This is the oxygen minus, 2 oxygen minus, this is phosphate group. Because of one phosphate group it is called as monophosphate, cytosine monophosphate, deoxy cytosine monophosphate. The structure will be like this students. Now fill the valency, all the valency first. So this is the structure of cytosine. So this is called as deoxycytosine monophosphate. So this way students I have drawn the two type of nucleotide. One is adenosine monophosphate where containing oxygen. Here there is a what? Deoxycytosine monophosphate, that is called a nucleotide students. So this is the nucleotide which is present in DNA, and this type of nucleotide is present in RNA. Understanding students? Structure of nucleic acid structure. Nucleic acid students, if you want to draw the structure of DNA, in DNA structure students, if you want to draw students, here the sugar I have drawn like this. This is the sugar molecules students. Sugar, this is the sugar students, sugar structure. Because DNA is a double helix structure students, you can draw the another strand of sugar like this. Now here these two sugar, they are joined with phosphate group. These sugars are joined with phosphate. Now here these students, these are the joined with the phosphate. Adenine with containing 2 hydrogen bonding with thymine. Cytosine combines with 3 hydrogen bonding with guanine. Okay students, so this way you can draw the nitrogenous base students. This is phosphate students. So this way, this is the nucleic acid structure students. Since it is a double helix structure students, these are the sugar monosaccharide and these are nitrogenous base and phosphorus. Understanding students? If you want to draw the structure students, so this way you can draw the structure of nucleic acid. The sugar strand I am drawing students. Since it is double helix, so this way you can draw the structure of DNA. Here these are the, if it is 3 dash, carbon number 3, so last will be 5 dash. If this is 5 dash, this will be the 3 dash. Understanding students? Now nitrogenous base you can draw like this. Adenine containing 2 hydrogen bonding with thymine. Cytosine containing triple bond with guanine. This way students you can draw the structure of DNA. Guanine containing triple hydrogen bonding with cytosine. Adenine combines with double hydrogen bonding with, so that is why it is called as what? Double helix structure. Understanding students? So this is the nucleic acid structure students, you can draw like this. So DNA double helix made by Watson and Crick model. silent. What are the silent features? Students, two nucleotide strand wind into right-handed double helix that I have already shown you. Two strand. This is the first strand, students, and this is second strand. So two polynucleotide strand wind into right-handed double helix. Two strand run in opposite direction. One will go on upward direction, other will downward. So two strand run in opposite directions. One form 5' to 3', other will form 3' to 5'. You can say 5' to 3' and 3' to 5'. This we can also read perpendicular to axis of helix. There is a perpendicular lines are there. They are going to form the perpendicular to axis of helix. Sugar, phosphate, backbone lie outside of the helix and bases lie inside. These are here the bases which are present inside. The hydrogen bonding between base of two DNA strands stabilize the double helix. These are, I have told, there is a hydrogen bonding, double bond as well as triple bond, triple hydrogen bonding. So hydrogen bond between base of two DNA stabilize the double helix. This gives rise to ladder-like structure of DNA as a double helix. Now here, students, adenine combines with thymine with two hydrogen bonding. Guanine combines with cytosine with three hydrogen bonding. So adenine, AT and CG, as adenine to thymine and cytosine to guanine are the complementary base pair. Two strands of double helix are complementary to each other. Understanding here, students? So this is the structure of single strand. If only one strand is there, students, 5' to 3', so that is called as that is always present in RNA. So this way, students, I have explained the structure of DNA double helix. This is very simple, students, for the examination point of view. If you like my video, please share it, subscribe it. Thank you very much. Have a nice day. God bless you.